Assalamualaikum and salam sejahtera. Today I will be presenting my final year project in the accumulation of heavy metals in Clariday species of West Peninsula Selangor. Starting with the background of study, heavy metal contamination in river endangers public water supply as well as fishery consumers. Heavy metals are a major source of aquatic pollution due to their bioaccumulation and non-biodegradable qualities in food. Clariday have evolved supplementary respiratory organs to survive in hostile environments with low oxygen and high toxic ammonia and high salinity levels. Consequently, they serve as excellent case study of the adaptation to terrestrial habitats. The West Peninsula region of Selangor, Malaysia, hosts diverse aquatic ecosystem, including water bodies where catfish are prevalent. Despite their ecological significance, there is a growing concern about the potential bioaccumulation of heavy metals in catfish tissue within this region. Heavy metals originating from anthropogenic activities and natural resources pose a serious threat to aquatic organisms and can enter the food chain, ultimately impacting human health. Therefore, a comprehensive investigation is essential to assess the bioaccumulation patterns and concentration of heavy metals in catfish tissues, providing crucial insights into the potential ecological and public health risks associated with the consumption of catfish from West Peninsula Selangor. The objective of this study is to analyze the accumulation of heavy metals concentration of zinc, lead, cadmium, arsenic, manganese, chromium, and copper of Clariday species from locations in Selangor, Malaysia using ICPOES. The significance of the study. This study serves as a warning for individuals who regularly consume fish, as even low quantities of heavy metals like zinc, lead, and cadmium can be harmful to human health. These metals can accumulate in fish tissues over time, especially in long-lived species like Clariday, which occupy higher positions in the food chain and are often sold in the markets accessible to the local populations. For the literature review, bioaccumulation is the process through which pollutants or metals enter the food chain and refers to the accumulation of contaminants in biological tissues by aquatic creatures from the sources such as water, food, and suspended sediments particles. The walking catfish is a freshwater fish that can breathe air and move about on land. It has evolved additional air-breathing organs and accompanying processes to thrive in such harsh settings. As a result, it is an excellent model for studying adaptations to terrestrial lives. Meanwhile, for the Clarias caripinus, the African Clariday is one of the most researched tropical Clariday in the genus Clarias. African Clariday has a strong genetically determined development potential, with a growth rate that is 3 to 4 times that of typical aquaculture species. Clarias caripinus is a highly appreciated and inexpensive staple in local communities as well as excellent aquaculture species. For the methodology of this study, in Kapar and Kuala Selangor, sample collection involved the separation of gill, bone, and tissue samples using stainless steel instrument, with two sizes categorized considered as small and big size, which is more than 30 cm and less than 30 cm. The collected wet samples were subsequently oven dried at temperature ranging from 90 degrees Celsius to 105 degrees Celsius overnight and stored refrigerated until further analysis. For sample preparation, 0.5 gram of dried samples from each organ were weighed and subjected to weight digestion. The digestion process involved heating the samples of 95 degrees Celsius on a hot plate. Then digested samples were transferred into 50 ml bottles sealed with parafilms to prevent contamination and stored in a cool environment to prior analysis. Sample digestion were performed using mixture of nitric acid, hydrochloric acid, and hydrogen peroxide acid. Finally, sample analysis was conducted using ICPOES to determine the heavy metal concentration of heavy metals. For the result and discussion, in Kuala Selangor, Clarias caripinus of larger size has the highest accumulation of concentration metal, which is zinc, which is less than 1 mg per liter for the three organs, which is tissue, gill, and bone, while the highest is manganese and metal accumulated mostly found in the bone and tissue compared to gill. However, in smaller size, Clarus carabinus accumulates higher metal compared to the largest size of catfish in the following species, with zinc accumulation more than 1 mg per liter in gills and bone. The second highest metal is manganese, mostly concentrated in the bone. Next, for Batracious species, size more than 30 cm, highest metal concentration is also zinc, followed by manganese, lead, chromium, copper, arsenic, and lastly cadmium as the lowest concentration. Heavy metal zinc accumulated highest in the tissue and gill as the lowest concentration. The highest accumulated of zinc in largest size Batracious is 4.1 mg per liter in tissue, which is quite high compared to the Garipinus species. Meanwhile, smaller size species of Batracious significantly accumulate less than 1 mg per liter of zinc, followed by other metals for all bone tissue and gill accumulated less than 0.05 mg per liter. Next is Kapar. For in Kapar, Clarus carabinus of larger size does not have that much variation, however in the bone has the most accumulation of metal such as zinc and manganese. Gill has the highest accumulation of every metal compared to tissue and bone. Next, for the size of less than 30 cm of carabinus which is zinc has the highest concentration, followed by manganese, lead, arsenic, chromium, copper, and cadmium. And the highest accumulation of metal is in bone, followed by gill and tissue. On the other hand, large Batracious in Kapar have very high metal concentration of zinc, which is followed by manganese. Tissue and gill accumulate more than bone of these metals. Lastly, smaller Batracious in Kapar have a very high metal concentration of zinc, which is above 2 mg liter, followed by manganese. Bone and gill accumulate more than tissue of these metals. According to the data, majority of the species and the locations, smaller fish seem to accumulate higher concentration than larger fish. The reason to this may be because of smaller catfish are often lower on the food chain and may consume food sources that are contaminated with heavy metals. As they continue to eat and grow, the accumulated of heavy metals become more concentrated in the tissue. In conclusion, the study has provided valuable insights into the accumulation of heavy metal in Clarus species, which is Clarus carabinus and C. Clarus Batracious, from two distinct locations, Kuala Selangor and Kapar, Malaysia. In summary, zinc has the highest concentration among other metals for both locations of Selangor, and manganese is the second highest concentration. Between smaller and larger size clarides, smaller size catfish have higher concentration. Between gills, bones, and tissue, the concentration values of metal vary for different species and location. Recommendations for future research include continuous monitoring of heavy metal levels in aquatic ecosystem, particularly in regions with industrial activities, to ensure the timely detection of any emerging threats. Additionally, further investigation into the specific pathway of heavy metal transfer within the food chain and their potential health impacts on human consumers would contribute to more comprehensive understanding of the risks associated with heavy metal concentration. So that is all for my final year project presentation. Thank you for listening.